Hi everyone, thanks for this opportunity. By Lab Roots, my name is Lan Hu. I lead the Data Science and Bioinformatics team at Viome Life Sciences. Today, I'll be talking about the metatranscriptome technology as an effective way to understand the oral microbial functional pathways, in addition to the microbiome composition, and more importantly, how to apply it in the context of a human health. My conflicts of interest. The, our human body is a walking ecosystem. A typical way of describing the human genome is that it encodes about 22,000 protein coding genes and many other non protein coding genes, such as link RNAs. But more importantly, our human body also hosts many invisible microorganisms, showing in the middle one. So, altogether, we have as many as the human cells as the microbial genes and cells. So together, the visible part and the invisible microbiome in that completes the human body in it and its health status. So we wanted to treat both sides nicely and keep a balanced human microbiome interaction. If it's out of balance, then we probably will be in a little bit of trouble. So, how do we keep it in balance? Well, the answer is by measuring gene expression or RNAs. So, in the left panel, you are seeing a side by side view of a healthy gum and inflamed gum from the same person. Now, the same person, the same DNA, it doesn't change, but the gene regulation and hence the gene expression does change. So RNA measures the actual health status, not just the potential. The middle panel shows the infamous P. gingivitis, which is often associated with the gingivitis condition. But it turns out, even though 80% and more people have it, only 15% express the toxin gingipin under certain conditions. So the presence of a DNA or the presence of this particular bug doesn't always mean that you have the disease. Most of the time, it's just happy to be here, just to hang out. So the right panel illustrates how measuring RNA could capture that relativity. So we quantify the expression level of various genes. Some are more expressed, some are less. As I shown in the previous slide, each person has more than 2 million genes encoded by the microbiome, but most of them are not actively expressed at any given time. Actually, only 5% or more, or less than 5%, are expressed at certain detectable level. So most of them just hang out and not necessarily doing bad things. In the field of microbiome, the technology has evolved over time. At the beginning, it's 16S sequencing, showing the first column. It sequences a fragment of DNA, which is 16S. By amplifying this particular region, we could we could detect what genus, and in some cases, even the phylum. So it's quite low resolution and they're definitely not measuring microbial functions because it's just one particular fragment of DNA. And that DNA it could come from even could come from dead microorganisms. So it really doesn't tell you what they're doing. And that they could tell you maybe who's out there. And then we evolved down to the metagenomics, showing in the middle column. This technology sequences the whole genome of the bugs. So then we could identify the particular microbes in higher resolution in the species or strain level, but it cannot detect RNA-based virus because we're only looking at the DNA. And again, it doesn't tell you the active activities. Then now to the right column, this is the technology I'm going to be focusing on. So metatranscriptomics, it sequences all the RNA molecules collected from the particular sample, and it's at a higher resolution, uh, just like metagenomics. It could detect the bugs at the species and strain level and from all live organisms because the dead ones they don't express. So by studying only live organisms, we actually will have a better insight into how they are impacting our health. And in addition to the bugs, we could identify the metabolites that produced or consumed from these activities. So metatranscriptomics, it tells us not only just who's out there, but also what they're doing. This is very important. Here is a high level illustration of using metatranscriptome technology to analyze RNA from a biological sample to results. It can be applied to many different sample types: stool, finger picked blood samples, or saliva, and others. The biggest challenge to work with RNAs is very high abundance of non-informative RNAs, such as ribosomal RNAs, shown in this red and green squiggle lines in the middle. So obviously there's a whole lot of red. And typically, like ribosomal RNAs, it could take up about 98% of RNA molecules from this pool. So we will need to deplete them to enrich the informative RNAs, largely from messenger RNAs. So from the many red to green, we wanted to get more greens, relatively little the red RNA molecules in the pool, as shown in the illustration. So after that, we could pull thousands of samples together to go through high throughput sequencing on the sequencer. And then the sequenced reads will be pushed to cloud to go through bioinformatics pipeline to do the uh, taxonomy and also functional annotation and identification, and then uh, downstream various AI machine learning models or other analyses. So I have mentioned that RNAs tell us both what the microbes are and what they do. So it is true that microbial composition can differ from population to population, from saliva sample to stool sample. But what we have learned from our metatranscript data is that the biochemical activities and the molecular functions show high similarity. Here's an example from over 2,000 people. Each person had paired saliva and stool samples, so they represent, in a degree, the oral microbiome and a gut microbiome. The left histogram shows the distribution of numbers of species identified in saliva and the stool. The saliva in green, stool in purplish blue. And so gut microbiome is more diverse compared to oral microbiome. Now the right plot is the distribution of the number of molecular functions annotated in KEGG ortholog. They show that's actually very similar. So we also did the analysis on what the functions were and their abundance, highly correlated. And if you're interested in learning more on this topic, the bottom lower side has a link to an article on this topic. The point here is that using microbial taxonomy compositions or species oversimplifies the difference compared to looking at the microbial biochemical activities. Now, we often say your eyes are the window to your soul. Now, the mouth is the gateway to your whole body nutrition and the health. So, the oral activity hosts a lot of microorganisms uh, illustrated in this cartoon. So, it might be a little bit unsettling to know what are out there. But the point here is that both the oral microbiome and the oral immune system, they defend against a vast array of the pathogens. Either one of them is impaired, we could have a whole slew of health conditions. For example, the oral cavity can support a pathogenic activity leading to chronic oral inflammation, toothache, all these kind of things. And digestion related health issues can arise, including putrefaction of foods and host proteins within the mouth. And showing the bottom, the halitosis, also known as bad breath, the periodontal diseases, and oral and throat cancers are just a few consequences of this impairment. Then, how do we apply metatranscriptome to learn more about these uninvited guests in our mouth and to deal with them if they are causing harm? So, we start with saliva, which is as non-invasive as it can go. So, you spit in a tube the first thing in the morning before brushing your teeth. And the saliva sample is a sequence, as shown in the previous slide about the workflow. And here is a graphic illustration of quantifying microbial functional pathways from the sequencing data. So from the left-hand side, we start from the FASTQ, the read files, the very standard output from a sequencing experiment. And then the bioinformatics pipeline would put the reads into different buckets and the genes, map to the genes, and annotate the microbial functions. And then we also do the normalization. So at the end of the second little figure, uh, we will get the relative abundance of each of these functions. And we use, again, KEGG ortholog or KO to annotate microbial functions. Here just showing two examples, and there are thousands of them typically in a sample, if not more. And the third one, the third panel, shows a highly iterative and a very involved process to select this subset of KOs that are collectively change, either upregulated or downregulated in the context of disease of interest. So at the end of this iterative process, we will get this winning set of KOs to represent this particular functional pathway. And then the last figure shows that we use a mathematical aggregation to combine the relative abundance of this winning set of KOs into one numerical value. It's a score between 0 and 100. 100 is the good direction, 0 is the bad direction. You know, a score between 0 and 100, it's a very intuitive. So score development is a very iterative process. And I think this is very helpful visual to put things into perspective. Imagine this super data cube, with the axis as a particular type of data. So we have the samples um, showing along the z-axis. So this is our development cohort, and we have about 10,000 here. And then there's the phenotype labels. It's about 500. They include demographic information, lifestyle data, health issues, any diseases the person may have, the drugs they may be taking, and a clinically validated questionnaire if qualified. And then we have another axis representing the RNA expression, the gene expression. This from the microbiome whole catalog, there are about over 100 million genes. Now, not all of them are expressed at any given time point, but this is the whole collection of all the data points. So the question is here, given this huge biological data set with more than just hundreds, millions, billions meaningful data points, how to identify and model a subset of the data points, however you want to slice and dice it, to capture and quantify the coordinated activity of RNAs that represent a biological pathway, function, or system. So this whole computational framework is published recently in a peer-reviewed journal. And I'm listing here, there's 5 steps summarizing this whole iterative process. From early on, the literature domain exploration to understand the score concepts, the phenotype labeling, multiple iterations, differential gene expression analysis comparisons between the case and control defined by any of these phenotype labels and to get the final winning set of KOs, and then the quantification or the aggregation of the relative abundance to represent this whole pathway activity. That's the score. So here's a little bit more detailed about the 10,000, around 10,000 reference cohort and the whole collection of oral pathway scores that we developed. Um, so the characteristics include the age distribution, the male female composition, BMI, ethnicity, tobacco use, brushing habit, flossing habit, um, and then the 8 uh, oral pathway scores here listed their um, targeted microbial activities and then the number of the KOs, the final winning set of KOs for each pathway scores. I'm going to focus on the 3 scores related in the oral inflammation pathways. So these 3 scores are represented in the middle table. And then on the left, it's showing some representative diseases or conditions caused by the oral inflammation. So we have a periodontal disease, that's inflamed gum, or receding gum, and the canker sore, and the dry mouth. You know, we all have one or the other at some point. And in the middle, it's a cartoon showing, in this between gum and tooth, there's a lot of things going on. There's a lot of molecules exist and a lot of microbes doing certain things. And on the left, on the right, top right, showing the key components that these pathways are uh, targeting. They include the sugars, the nucleic acid, proteins, protein glycans, and the glycoproteins, the metabolism of all these various compounds, and the key functions related to the oral inflammation. So now let me come back to the 3 scores. The oral LPS biosynthesis pathway score is designed to typify inflammatory activities, specifically highlighting contributions of a detrimental gram-negative biofilm. The middle one, oral periodontal mucin degradation pathway score, is focused on salivary barrier defense, which preserve the health of the oral activity. And the mucin degradation is a known factor in periodontal disease. The third one, the oral periodontal pro-inflammatory pathway score, is heavily focused on both inflammation and periodontal destruction, which are defining symptoms of gingivitis and periodontal disease. So after designing the score, they need to be validated. And here is a validation result. The scores are validated on the large independent saliva sample cohort, about 15,000. So this is independent from the 10,000 that I showed before. It shows that the relevant oral pathway scores are significantly worse in individuals with gingivitis, acid reflux, and the nicotine addictions compared with controls. There's a lot of information on this chart, so let me focus on the gingivitis results first. The bottom, the numerical value 0 to 100, that represents the score range. And the x-axis is showing all the 8 scores. And the red is the case, meaning people report to have gingivitis. And the control, what is control? It refers to people who do not report any of these conditions. And we match these two groups by age distribution, the male female composition the breakdown, and also BMI distribution, and also in 1 to 10 ratio, meaning one case we will match 10 control individuals from the pool. So the red and the blue, the bars showing the dot in the middle, that's the mean, and the plus minus standard deviation of the score from two groups. And these 3 oral inflammation pathway scores indicated by the arrows, showing that they are significantly worse meaning lower in case relative to their control counterparts, and they have a different levels of significance. Now, if we look at the acid reflux and the nicotine addiction to other conditions, the story is the same. And actually, look at the middle in acid reflux. All 8 scores are significantly worse in control, in case than in the control. And the nicotine addiction, the same story here. So with that, I would like to summarize what I have talked in this presentation. So I explained the metatranscriptome is effective to measure both microbes and their active functions. And we designed the scores to quantify oral microbial functional pathways using saliva metatranscriptome data. To apply these scores and the metatranscriptome technology, we also report that these pathway scores are significantly worse in individuals with gingivitis and other conditions compared to their controlled counterparts. So, with that, I would like to conclude my talk. And thank you, everybody, for your attention.